Hi, my name is Lacey Novosel. I'm a senior biology major.
Hi, my name is Katie Park, and I'm a junior biology major.
And hi, my name is Nathan Smith, and I'm a senior biology and public health major. Today we'll be talking about classical conditioning and Aplysia californica associated with learning using light and tactile stimuli.
So what are Aplysia californica? Aplysia are commonly referred to as sea slugs or the California sea hare. They are found off of the coast of California and northwestern Mexico. Aplysia inhabit the photic zone of the ocean, which is where they graze on algae, their main food source and what we feed them here at QU. They don't go any deeper than 18 to 20 meters. Aplysia have a relatively short lifespan of only like 18 weeks normally, so we get them as young adults here at QU to train them. Aplysia are hermaphroditic, meaning they have both male and female reproductive parts, and that's actually why we keep them in separate tanks. And Aplysia have a few defense mechanisms, such as inking and balling, which we've studied here in the past at QU.
Aplysia have clusters of neurons that create a ganglia, and this structure controls the core forms of learning, such as habituation, sensitization, and classical conditioning. Aplysia demonstrate this through simple reflexive withdrawals. Now, humans have around 86 billion neurons with an amygdala to create a complex neural system. Despite these neural differences, both Aplysia and humans rely on synaptic plasticity to strengthen associative responses. And although Aplysia lack an amygdala, their ganglia exhibit fundamental mechanisms of learning that are parallel to the human basic processes, the human neural basic processes.
So as many of you know, we've done a lot of previous research with Aplysia here at QU, and a lot of that has been classical conditioning research, and we have actually been able to prove that Aplysia have some capacity of learning and memory, where we were able to classically condition them to things like frequency, brush stimulus, and poke stimulus. So this semester, we kind of wanted to shift that research in a different direction, which is a focus on fear conditioning. So with this, we were going to test the ability of Aplysia to see their fear responses to different stimuli.
So we asked, can Aplysia develop a conditioned aversive response to a previously neutral or positive stimulus? If so, this would demonstrate how activity-dependent synaptic plasticity remains evolutionarily conserved across all species. With this, we can bridge the gap between simple neural circuits and more complex systems in humans, exemplifying how repeated experiences lead to modified neural connections. Furthermore, with these biological mechanisms can provide insight into how psychologically, neurologically, and physical connections can lead to adaptive behavioral responses to stimuli. In humans, this could explain how we develop conditioned fear responses, such as phobias and other subconscious processes and reactions to stimuli. So to begin our research, we outlined two questions. One, will Aplysia develop a conditioned fear response quicker to a neutral stimulus when paired with an aversive stimulus, or to a positive stimulus when paired with an aversive stimulus? And two, will Aplysia trained with 5 conditioning trials show a stronger and more persistent reaction than Aplysia trained with only 2 trials? We hypothesized that our brush stimulus would show faster conditioning than our light stimulus due to it being a touching sensation.
So this is just going to walk you through how we set up our rooms. So we have 4 rooms to train the Aplysia with 3 Aplysia in each room, all individually tanked. We tested them on Tuesdays and Thursdays, carrying Monday, Wednesdays, and Fridays. So we wanted to have 2 rooms dedicated to the light and 2 rooms dedicated to the brush. Essentially in the light room, what we did was we laid the Aplysia in a pan. We would shine a light at the end of the pan with all of the lights off in the room. And Aplysia's natural response is to go towards the light. After this, we would administer the poke. And then we would repeat it for 2 trials in one room and for 5 trials in the other room. And then the same for the brush. We would place the brush on the Aplysia's back for an allotted amount of time. Then we would administer the poke. And again, we had a 2-time trial room and a 5-time trial room to do what Katie was saying, test how frequently we needed to be doing this to the Aplysia. So classical conditioning, just to cover our parameters here, our unconditioned stimulus was the tail poke, with the unconditioned response being the withdrawal. So essentially when we would poke the Aplysia's tail, they would retract it. We do have some videos here to show you shortly. The positive stimulus is the light, meaning, as I said, naturally, aplasia, if you place a light in front of them, they're going to go towards it. And the neutral stimulus for aplasia is also the brush. So if we place a brush on the aplasia's back, they have no response to it. Our conditioned stimulus is the light or brush after pairing, and then the conditioned response, eventually, the goal was for that to be avoiding the light or withdrawing to just the brush alone.
So before we could continue our experiment, we needed to establish the aplasia's initial responses to our stimuli. In this video, you can see that the aplasia is moving positively to the light, which is what we are needing. And we tested this twice with normal rest days in between to ensure it was a consistent response. And in this video, you can see for our tactile stimulus, we gently placed the brush on the backside of the aplasia, and it administered no response recorded, making it our neutral stimulus. And then in this video, we showed how we used a needle to poke the tail of the aplasia to administer a tail tuck response. This is going to be our goal conditioned fear response.
Okay, now that we've covered what our positive and neutral stimuli were for classical conditioning, we'll dive right into our data, starting off with rooms 1 and 2, our light stimuli training. As you can see, from right here, room 1 was trained twice per session to our light stimulus, and room 2 was trained 5 times per session to our light stimulus. Across 16 testing dates, we trained aplasia with these training paradigms with a naturally positive light stimulus to condition our adversive tail tuck response. As you can see from the line graph from the collected data above, the 5-time training paradigm produced the quickest conditioned response on March 5th and an overall number of conditioned responses to our light stimulus throughout our 16 paired training days. Now, we can simply look at the data, but we also want to understand the statistical significance behind this. To understand the statistical significance, we ran a Wilcoxon signed ring test to analyze whether increasing training intensity from 2 times to 5 times a session significantly affected tail tuck responses. A Wilcoxon signed ring test essentially tells us, across all my paired observations, does one condition tend to produce higher values than another? Because our data are paired by date, the Wilcoxon signed ring test is more appropriate than a paired t-test, which just sums up our averages. So after running the Wilcoxon signed ring test, it revealed a significant increase in tail tuck responses to light stimulation under our 5-time training paradigm compared to our 2-time training paradigm. It produced a W score of 9 and a statistically significant p-value of about 0.027, which is far below the significant value of 0.05, indicating statistical significance. This led us to the conclusion in our light stimulation rooms that increased training intensity enhances classical conditioning in Aplysia californica. We did the same thing with our tactile stimuli training rooms, which is our brush stimulus. Room 3, much like room 1 aplasia, were trained twice per session, where room 4 aplasia, much like room 2 aplasia, were trained 5 times per session. Across these 16 testing dates, we compiled our data and put them in this line graph. The orange represents our 5-time training paradigm, and the blue line represents our 2-time training paradigm. As you can see, once again, our 5-time training paradigm produced the quickest conditioned aversive response to a neutral stimulus, which is our brush, and overall it conditioned the most amount of responses, developed the most amount of conditioned responses across our Aplysia californica. Again, we ran a Wilcoxon signed ring test to understand the statistical significance behind this data. After running the test, it revealed an even greater significance in tail tuck responses to tactile stimulation versus light stimulation under the 5-time conditioning paradigm compared to our 2-time conditioning paradigm. It produced a W score of 9 and a statistically significant p-value of about 0.020, which is far below the statistical significance standard of 0.05. Again, this led us to the conclusion that increased training intensity enhances classical conditioning in Aplysia californica. Finally, we wanted to compare the differences between the light stimuli, our naturally positive one, and tactile stimuli, a naturally neutral stimuli, to see which one can classically condition an aversive response, a tail tuck, most effectively. As you can see, we compiled all the data into room 1 and 2 being our light stimulus on the blue line, and room 3 and 4 being our tactile stimulus in the orange line, and we put it in a line graph. From the data, we can see that the tactile stimuli not only produced the quickest condition response, but it also produced the greatest amount of condition responses when classically conditioned our Aplysia californica. It is important to note, however, that within our last 4 days of training, we started seeing more consistent responses with our light stimulus trained Aplysia for classically conditioned and their conditioned responses. Finally, we ran a third Wilcoxon-Stein-Ring test to compare whether light or tactile stimulation produces a stronger tail tuck response overall. After running the test, it revealed that there was no significant difference in tail tuck responses between a positive or neutral stimuli, even though it was observed through our training above, just observed by us. It produced a W score of 35.5 and a statistically insignificant value of about 0.28, which is far above the statistical standard of 0.05. This led us to the conclusion that there's no significant difference in training with a positive or neutral stimulus for classical conditioning with Aplysia californica across our 16 trial days, while further research and more training could produce different values than we observed.
So some of the major takeaways from all of that data that Nathan just gave us is that classically conditioning did occur here and Aplysia are capable of it, but it really depends on training intensity. So for both rooms, increasing that 2 times to 5 times was significant in increasing the response from our Aplysia. And it's also important to note that while the results did show that the neutral stimulus conditioned an aversive response more efficiently than the light, it is not statistically significant. So we did observe that, right away, those first couple trials, they are responding to that brush a lot more, but that statistical number just isn't there yet. It's also important to note that it did take longer for them to be trained to something that they were initially positive towards compared to something that was neutral, which was the brush.
To conclude, we did prove that Aplysia californica can show associative learning through fear conditioning. To improve this experiment in the future, we would recommend to increase the number of training days as well as keep at least 5 trials across all rooms. We could also reduce the stress given to the Aplysia through gentler handling and consistent poke intensities. We could have also added a true control group, meaning that there would be no poke, just the light or tactile stimulus. For quantitative data, we can make it more precise by counting the percentage of conditioned responses per session and as well noting the latency to respond. For our future research using similar conditioning techniques and optimum training times, we will test if Aplysia can correlate a positive response to an initially negative needle poke. Positive conditioning could correlate to how humans are able to overcome phobias and other subconscious fears. Using the help of AI, we will have it predict expected and extended data to compare our future findings to.
We would like to give a special thanks to the University of Miami for providing us with our research animals, our Aplysia californica, and their food, Dr. Kimberly Hale, our faculty mentor, Grace Welch and Andrew Lichtenberg, our fellow research members, and KU Maintenance and KU Security for providing appropriate training environments. Here are our references.